glioblastoma, and it will be given by Professor Kang from Seoul.
Good morning, ladies and gentlemen, and I'd like to talk about the albumin as a drug delivery system. I'm a nuclear medicine physician, and so I can do multimodal imaging for the biodistribution and targeting efficiency. And so I have many chance to test in other nanoparticles such as silica, gold nanoparticle, silver, and supplied by the other material scientists. But the problem is that the molecule and particle is very new. It is very difficult to go translate it into the clinic. And so I turn back into the very traditional, in human bodies, natural nanoparticle. And so it is also already the first publishing for visualizing the EPR impact was albumin as well. And there are also one of the FDA approved drugs is albumin. Because an albumin is constituted of 60% of total plasma rate and the circulating long time like antibody, because of the rescued by the FcRn, it is almost the same mechanism of the antibody rescued. And the antibodies, albumin, engulfed by the hepatic cell as well as the endothelial cells, but rescued again and coming back to the plasma. And that's why the albumin circulates for a long time, not like the other, and the ribosomes are sequestrated by Kupffer cells, and also very soluble. And it already acts as the natural drug delivery system. Many hormones and ibuprofen and the diazepams are binding to the albumin and the circulating, and that's why the kind of drugs there is a long plasma half-life. And also, in nuclear medicines, we already used technetium albumin scan in human for decades. And this is one of my patients in this imaging of the, where's this one? Oh, I'm sure. Okay, and from 5 minutes, okay, 5 minutes up to 24 hours. And the albumin is generally located inside of the vessel. And so you can see the major vessel, heart over here, and the liver is also hypervascular and the organs and the major vessels here. And after that, 24 hours, this patient has a protein losing enteropathy. So there's some leaky and albumin leaked out through the bowel. It is not an unusual one. It is not an unusual one. Generally, it should be like this. Circulating a long time. But when I'm looking at the albumin, everybody think about this is on the EPR effect, but I accounted in this article, and the albumin, serum albumin, maybe are just a SPARC and the GP60, other maybe other and the active transporting systems may be related. And so we tested it in many different cell lines, and some cell lines, especially glioblastoma cells, U87Mg, and this is a glioblastoma cells from the patients in my hospital, and it shows a high expression of the SPARC. SPARC is a secretory protein, acidic and rich in cysteine, and also Western blot shows almost the same. So we want to test what is the SPARC mechanisms of the albumin transport. And so we made a knockdown cells of the U87Mg and the SPARC knockdown and native cell. And also we tested in vitro fluorescence imaging and the uptake of the albumin. Albumin, serum albumin is the site 5 dyes attached on it. And so this is a control, and the U87Mg and the SPARC excreted in the SPARC knockdown one. And so there is albumin uptake inside of the cell. But when we increase and externally treated the SPARC, increase the dose, and so you can see, and even the knockdown cells engulf the human serum albumin. And so SPARC may be related in endocytosis of serum albumin into the tumor cell. And then we did an in vivo fluorescence imaging, and we inoculated 2 different tumors, and U87Mg over here on the right side, and the knockdown, SPARC knockdown cells on the left side, and almost the same size. But after the 24 hours, and there's much more uptake over here only in the SPARC positive cell. It means that the human serum albumin accumulate more in the SPARC positive. And also, we did test, and the dextran, almost the same size, And this is human serum albumin just around the 66 kilodalton size. And we did test the dextran at the 70 kilodalton size. Dextran is just the EPR effect. There's no target in our the system, in the CLA system. And so in that case, dextrans are SPARC knockdown. You can see the difference, slightly different. And the SPARC knockdown cells are less uptake. But if you're looking at the human serum albumin, and the SPARC knockdown cells almost more than double. And so the SPARC is maybe related, not just the EPR, not just the open the more of the endocytosis gap. And also we did the ex vivo fluorescence imaging and take it out tumors, and we see the dextran distributions, and as well as the human serum albumin distributions, and showing CD31 is a vessel and the cilia cell. This is the SPARC native one, and you can see the dextran distributions are like here, but human serum albumin is distributed, and throughout the all here surrounding, and they're engulfed by the inside of the cell. But if you're looking at the SPARC knockdown cells, and also there is a dextrans extravasated outside, but human serum albumin mostly within the endothelial cell, and some of that is extravasated much less. And so maybe in the SPARC related with the receptor-mediated transcytosis, but we don't know yet and what kind of receptors are mediated. And then we're trying to use therapeutics as well. This is a talk about the theragnostics. In nuclear medicines, we can change, if we change into the different radioisotopes, such as the lutetium-177s, and both emits and beta ray and gamma ray at the same time, and we can get imaging and treatment as well. And so I'm going to show you, and this is playing, not playing, okay. Lutetium-177 is emit both beta ray, gamma ray for the imaging, and beta ray is electron, is destroy the surrounding 1 millimeter, just around 1 millimeters. Maybe attaching and conjugating into the peptide, the protein albumin, or the nanoparticles. And if we injected that, and then it's circulating in our body, and finally, they're going into the target. And the beta rays and destroying the surrounding cells. Even if it's not attaching to directly the cells, and it emits in the surrounding 1 millimeters, and 1,000 cells die. So the accumulation is very important for this kind of strategy. And so going to the endocytosis, and the radionuclide remain inside of cells until the cell death. And the gamma rays are coming out, and transparent through the body, and finally the SPECT imaging systems detect the distribution of the drug itself. And so we radio-labeled the lutetium-177, the human serum albumin, in the mouse model. And then this is a control, and this is a different dose, and the 0.5 millicuries and double dose. So it's suppressed. But the problem is that the higher doses kill the body itself and the animal. And we also get imaging from using gamma ray from the human serum albumin distributions. We can see the tumors over here, and follow up serially and up to 5 days. And albumin is accumulated first, and the 4 hours and 1 day, but finally disappear. And so you can see the major vessels are also disappear. And this is one of my strategies, using albumin as a platform for the imaging and the therapy as well, using the lutetium or therapeutic and diagnostic radiopharmaceutical. And we can change the albumin and maybe attaching the HER2 aptamers on the surface that we are doing before, and folic acid, and we can target on different kind of cells. These are our teams. We have physics and chemistry, molecular biology teams together working, and most of this work was done by this and Professor Song, our research professor. Thank you very much for your attention.
Thank you very much for the very interesting talk. There's a question in the back, left, there.
Thank you. Very interesting talk indeed. You mentioned that you also use patient-derived lines. So is the expression of SPARC similar to U87s, and in that context, were the in vivo models using these patient-derived lines?
Yeah, so actually, we didn't do the patient-derived xenograft yet, but we are going to try the in-human. Actually, technetium-albumin scan is already available in clinic, and so we already approved by Institute Review Board and we are going to get imaging of the human and the brain scan using the albumin. This is my plan.
There's another question over there.
Yeah, question. Yeah. Have you tested in an orthotopic glioblastoma model?
Oh, not yet. That's the problem we have, and this is only xenograft models, and so that's why we want to do that, and the technetium albumin scan in human directly. Yeah.
Other questions? Okay, then we close here, and the next.